So, my great pleasure today to see my graduate student defend a thesis. Christine grew up in the mountains of Tennessee, Knoxville. She just made me sure it's not Nashville, it's Knoxville to say, so it's two different things.
It's not Louisville either.
It's not Louisville either, and it is not Memphis, Tennessee, where I grew up, based on my strong southern accent. At least scientifically, I spent 8 years there. So Christine went to McAllister, yes? I don't know the small American colleges names well, but it's a great college, of course, in Minnesota. And she came here for graduate student studies, and I was very pleased that she really wanted to find someone from Tennessee to do the graduate work. You know, in Tennessee we're playing your songs, come to Tennessee. So we played the songs together with Christine for several years now. And she's an amazing singer, I will tell you. She's very imaginative, she's very persistent, and you could tell this based on her work. Not every project, every good project doesn't go very well, as I know. Only bad projects go very smoothly. And we had lots of hiccups and problems, as you would expect with anything. And it's amazing how Christina suddenly were capable to come up with very unusual decisions. And she truly amazed me quite a few times during her graduate work. With all this said, it's my great pleasure to give the podium to Christine Lloyd. Please. Continues mic.
So, should I be mic'd, or am I projecting well enough as it is? All right, excellent. I do not want to have to deal with feedback. So, thank you, everybody, for being here today. This is a little bit surreal to see years of my life be turned into a 1-hour presentation. I can only imagine how bad it is for people who have been doing research for 20 years and are trying to sum up the last decade of research in a 1-hour presentation. But so, the very general question that we started out with is, what does E. coli natural variation tell us about how these organisms become pathogenic? So, E. coli adaptation to the extraintestinal niche, using natural variation to explore mechanisms of pathogenesis. And I apologize, I was practicing my Slavic accent, but I couldn't get it quite right, so unfortunately, it isn't going to be like your usual presentations from the Sokurenko lab. All right, so first off, just quick refresher for people on what is E. coli. So Escherichia coli is a pretty much universal member of the human gut microbiome. There are a great many of these strains that are commensal, and so these help to inhibit pathogens being able to colonize the gastrointestinal tract. They also provide vitamins to the host, so they're good actors. However, there are a great number of strains as well that are pathogenic or have pathogenic potential. So how does E. coli get around? Because obviously we don't all have our own E. coli forever. So E. coli is transmitted through fecal-oral route, which basically means it gets ingested somehow. It goes on to either just pass through the host or persist, multiply, and then have more of itself to pass on. And then when it's passed into the environment, it may be able to transmit to a new host. So disease is caused by E. coli. People are probably familiar with diarrheal disease being caused by E. coli. I'm sure most of you have heard of the 0157H7, which is one of those nasty ones that makes the news a lot. But E. coli is also capable of causing a number of diseases outside of the intestinal tract. So urinary tract infections are the ones that probably the most people have experience with. About half of healthy women will have a urinary tract infection before menopause, and the vast majority of those are caused by E. coli. And there's also a number of other ones. And all the extraintestinal niches, when we look at these bugs, we can't look at it and say, oh yes, this is a meningitic strain, or this is one that came from UTI. We sort of lump them together as XPEC. So how does transmission work for expat strains? Because again, we're talking about outside of the intestines. So how are those getting transmitted? It actually starts out very much the same. These are ingested. These may pass through the gut or persist in the gut. But then these E. coli from the gut will go on to infect an extraintestinal niche, the urinary tract, the blood, etc., from within that host. And in the case of urinary strains, at least, these can then go on to re-enter the environment and potentially infect new hosts. But you notice that this is going to be a case of they are subject to the selective pressures of the intestine and then of the extraintestinal niche. And then if they go on to transmit further, again, the pressures of the intestine and the pressures of the extraintestinal niche. So how are they managing to adapt to this? There's a number of mechanisms through which E. coli, or really any pathogenic bacteria, is going to be able to go from being in its commensal compartment, being fat and happy. And we're using as an example an adhesion in this because this is what has been studied a lot in our lab and is fairly well understood. Though, as I'll tell you later, there's still more for us to learn. And so in the commensal compartment, it will have some adhesion that fits really nicely in here. But if it goes into a virulence compartment, say the urinary tract, the adhesion isn't really doing its job very well. It's not going to stick. It's not going to be able to stay around. So how does it manage to stay around? It can get new genes from outside. So now it has a new adhesion. And what happens a lot of times in these cases, or in a number of cases, there's going to be the fact that what was the old adhesion or old gene that was good for the commensal compartment isn't so great for the virulence compartment. That may be lost entirely. It might also be that this bug could increase its expression of the adhesion. So while the affinity of the individual adhesins isn't that great, when you get more of them being expressed, on the whole, bacteria is better able to hold on here. Or you could get protein mutation, where it's the same gene, but now characteristics of the protein have changed such that it's actually able to persist in its virulence compartment. So we took a couple of approaches to looking at how virulence is evolving in these E. coli. The first one that I'm going to talk about is sort of a genotype to phenotype approach. So, as I'm sure everybody's familiar with, we've got a lot of gene and genome sequences that are available now. There are more of them being published all the time, and it's this wonderful, massive amount of information, but you have to actually figure out how you're going to dig through it before you can find something interesting. So, looking at the available sequences that we have, and then pick something that's interesting, something that we think has a likelihood of being involved in adaptation to this pathogenic compartment, and look at what variations are going to be present across all of these genotypes. That's potentially interesting, but where I think it's really neat to look at is what are these variations associated with? Are they associated with some characteristic of the organism? And we can also go in the reverse direction. We can start out with a phenotype that we're really interested in. So in this case, we've got urinary epithelium that's being shown up here on the left and right. And on the right, we've got bacteria that are really good at binding to the urinary epithelium, as opposed to our strains that are not. So we've got a phenotype of interest. And in these days of cheap genome sequencing, we can sequence the entire genome, or genes of interest, and look at what changes are associated with this big phenotypic shift that we see. So the first thing that I'm going to talk about is going from genotype to phenotype. So what is our area of interest? Because obviously we can't look at the whole genome. So our lab has for quite a while been studying the E. coli type 1 fibrid. So these are these hair-like projections on the surface of E. coli, and very much the majority of them have these, with, at the very tip, an adhesin protein called FIMH. FIMH is responsible for binding to monosylated substrates, so this allows these bugs to bind to human cells. This is encoded by this FIM operon. We've got a couple genes and an intergenic region that are involved in the regulation. we've got the major subunit, present in hundreds of copies per fimbrial, and we've got the adhesive, which is responsible for the binding phenotype that we see. And this is what's been the most studied about type 1 fimbrial operon. And it's sort of an example. So this has been seen in the lab, that you can start out with a fecal-like FimH, so something like you would find in a commensal strain. These organisms are not really very well able to bind to urinary tract epithelium. It's not what they're good at. With a single SNP, it's possible to now have bacteria that bind with very high affinity to the proteins found on the urinary tract epithelium. And this is seen over and over in UTI isolates of E. coli. Many different SNPs that will all, with just a single change, allow these bacteria to colonize the urinary tract and cause infection. But we've got like the whole rest of the operon to look at. So what else in this operon could be interesting? Yes, we've seen this one very dramatic phenotype, but it's unlikely that that's the only thing that's going on when you have an organelle like this that's so important for pathogenesis. So again, we have many published genome sequences. We're looking on the order of 1,000 was what a lot of this research was done with. And in this case, we did nucleotide blast for the fimbrial operon. Now, the majority of the published genome sequences that are out there are available as contigs. So not a complete circular genome, but many fragments. So then it's necessary to go in manually and actually assemble a full operon, because in a lot of cases, the operon was split across 2 or more contigs. Once that was done, we could then go in and look at what SNPs were present and were these associated with a phenotype of interest. But let's step back. So what are the genomes used for analysis? Again, we've got a whole mess of these, and this, as you can imagine, the sample set is a bit biased by what the people with the sequencers are interested in. So there are people who are very interested in diarrheagenic E. coli, so we've got a lot of samples from diarrhea. We've got a lot of samples from healthy stool. We've got a fair amount of laboratory strains, some environmental, and a decent handful of expecs, as well as quite a few that were unspecified. So what's the overall picture of variation? Total variation by gene is pretty much what we would expect to see. These genes are generally conserved, with the exception of the major fimbrial subunit. So again, remember, this is the one that's present in many, many copies per fimbrial. This means that it's a target of the immune system, and so there's strong positive selection for variation on the solvent-exposed portions of this protein, as well as a fair amount of recombination that happens here as the bacteria are trying to avoid the immune system. This variation is predominantly synonymous. Again, this operon is overall conserved, with the exception, and even in FimA, there's a whole lot of synonymous variation going on. Overall level of non-synonymous variation is very low. Again, this is all what we'd expect. This is all what's been published before. But we also looked at the non-coding regions. So, looking at the operon, and all these non-coding regions upstream of the genes. What's the variation that's happening here? Interestingly, it actually seems to be fairly conserved. The level of variation seen on most of these genes is lower than what's seen in terms of synonymous variation. And I was also really interested to see that the region upstream of FimA, we're not seeing greater variation than in the other genes, unlike the coding variation that we saw with FimA. However, what we do see is an increase in non-coding variation upstream of the adhesion, upstream of FimH. And of course, the first question is, so this dotted line is the average level of synonymous variation in genes of the operon excluding FimA. So the first question is, well, this could argue that the non-coding region is conserved, except for at FimH, where the selection pressure is just relaxed. And the other question that we had was, all right, this doesn't look like the upstream variation is varying, sort of co-varying with the coding variation, as in the level of variation doesn't look like it's being carried along by the level of variation we see in the genes themselves. And no, we don't see that. There's not any predictive power of the variation within the gene, either synonymous or non-synonymous, and the variation in the non-coding region. So this probably isn't just some sort of holdover. So let's look in a little bit more detail with what's happening upstream of FIMH. So 19 nucleotides between FIMG and FIMH. We do see 5 polymorphic positions, and 3 of these are all present within a single branch. So this change happened once, and it went on through the related, strings. However, this change from G to A at position -15 relative to FIMH doesn't have such a simple inheritance pattern. This may be that the change happened twice, or that the change occurred and then occurred down here, and then reverted and was reacquired. So this makes you wonder, is there some sort of selection for this variation? Or is this somehow important in pathogenesis? So this nucleotide is what we call a hotspot. This is something that we've done a lot of work with amino acids in the lab. So hotspot amino acids, where you have, for example, a position, 27 here, and you either see the same mutation occurring twice independently of each other. Obviously, you have to consider recombination, because recombination will give you false positives. And yes, we did work to make sure that we were not getting false positives from recombination. Or that the same position is selected for variation. So instead of being the same change, 2 different changes at the same spot. So of course my first question was, knowing the strains as I did, I noticed that this particular polymorphism seemed to be associated with strains of expac origin. So this means UTI, sepsis, meningitis, etc. So what I'm trying to show you here is these little circles are representing, at each branch, what fraction of the strains there are expac versus not expac in origin. And just at first glance, these branches that have the -15A polymorphism do seem to have more expacs, particularly this branch. And while there's plenty of expacs that don't have this variation, there is a significant association between the 2 of these. So what does this mean? What is this polymorphism potentially good for? Well, one of the big things is that the predicted ribosome binding site for FIMH is immediately adjacent to this site, and could possibly include it, though that's not the prediction, but it is, again, we've got purines right next to the ribosome binding site. It does make me wonder. There's a little bit of work that I did that indicated that this polymorphism could affect the mRNA secondary structure. This was interesting, this was suggestive, but it was very dependent on the conditions that were used for the prediction. And so this is something that would have to be verified with some actual in vitro work, looking at mRNA folding with and without this polymorphism, and what the effects of this are on mRNA stability, translation, potentially other effects as well. All right, so what can we say here? Well, overall, non-coding variation over most of the operon is indicating conservation. So this does suggest that these non-coding regions are good for something. They need to have a specific function. The non-coding polymorphisms upstream of FIMH, at least one of these suggests positive selection, again, pointing to a functional role of these non-coding regions. We're also interested, again, remember that expac is having to deal with these alternating selection pressures, the selection pressures associated with the gut, and then with the extraintestinal compartment, with the gut and the extraintestinal compartment. So this overall analysis that we did of all of these strains, the downside is it lumps together a whole lot of strains with a whole lot of different pathotypes. Obviously, the selection pressures for diarrheogenic E. coli versus urinary tract E. coli, completely different. So how do we go about teasing out the most recent changes? Because since we're interested in the expac niche, the most recent changes in those strains are likely to be of the most interest, because these are likely to be associated with how these strains went from being commensal to being a problem. So one of the big accepted ways to do this with E. coli, to figure out what strains are the most closely related, is multiple locus sequence typing, or MLST. So this uses the sequence of 7 genes that are arrayed around the chromosome. These are chosen because they're very well conserved. These are housekeeping genes. And because they've been shown to have low recombination. So these should be getting vertically transmitted as opposed to horizontally transmitted through E. coli. And we looked specifically at sequence types. So a sequence type is defined as a group of strains with identical alleles at each of the 7 loci. These have been shown before to generally be fairly closely related. So for example, one little branch of the MLST tree for the whole set, see a number of different sequence types branching apart from each other. We'll just look at one, ST10. This includes some of our really, most beloved lab strains, for example. So how variable is the Fim operon within an ST? As it's been shown before, pretty darn variable. Just because organisms have the same sequence type does not mean they're going to have the same Fim operon. And just because organisms share the same Fim operon does not mean they're going to share the same sequence type. Fim operons are commonly sort of traded between E. coli as a whole. So looking simply within a sequence type isn't going to show us the most recent variation. Instead, we're going to look at these sub-ST clades that are appearing. So within closely related strains, very closely related Fim operons. So looking across all the sub-ST clades that were present in the set, combined all the variation that I saw over this very short period of time. It's a little hairy, though you may notice, so blue is indicating that it's a synonymous change, red is non-synonymous, and black is intergenic. Apologies, let me turn down the lights, I forgot to do that. This will probably be a lot more visible now. So hopefully you're noticing that there does seem to be rather a lot of non-synonymous variation going on. But, okay, is this really anything that's interesting? So on the left, we're looking just within the same ST. So these only have the same sequence type. We're not saying anything about how closely the operons are related. And we're seeing about what we would expect. Less non-synonymous variation than synonymous variation. However, when we look within the sub-ST clades, the overall level of variation is, of course, much less because we're not trying to compare relatively unrelated Fim operons. But, in fact, non-synonymous variation is much more common than synonymous variation. So we're dealing with an estimated DNDS slightly over 1.
Is this,
Yeah.
could you just go back to, what's the vertical axis here?
The vertical axis is number of mutations seen.
So it's the number of SNPs within each of those contexts.
It's the number of SNPs seen in each of those contexts. Yes, thank you. And again, we had previously seen this case of the non-coding variation looking like it was happening more often in H. So you're, considering, does non-coding variation also show some difference in pattern in the most recent? Certainly worth looking at. In fact, when we look at the same ST, we're seeing, about the same levels of non-coding versus coding polymorphisms. So this is the fraction of the sites of a given category that are polymorphic. Whereas when we look at the sub-ST clades, we're now seeing a switch from slightly more coding variation to slightly more non-coding variation. So what is this suggesting? Well, this does suggest that there is increased non-coding variation over the short term, and that the sub-ST clade variation shows a strong tendency towards non-synonymous and non-coding SNPs. So again, this idea keeps coming up that, yeah, non-coding regions are doing something. And this suggestion that over the short term, positive selection may be dominating. So let's go on to the other approach that I mentioned, the phenotype to genotype approach. So again, we start with our two interesting phenotypes, whatever they are, do genome sequencing, and then look at the differences we see between these strains that are specific to the phenotype. We were actually lucky enough to be able to work with a really interesting set of bacteria. So these are actually from the lungs of patients with cystic fibrosis. Cystic fibrosis is caused by defecting chloride transport, which is throughout the body. It's present in about 1 in 3,000 live births in the Caucasian population. And it results in substantially reduced life expectancy, which is primarily due to pulmonary infection. So they have problems with nasty, thick mucus in their lungs. They can't clear it out. Pathogens stay in the lungs. You get very nasty infections. The pathogens that have primarily been studied for cystic fibrosis are Pseudomonas aeruginosa, Staphylococcus aureus, and Burkholderia cepacia. Pseudomonas aeruginosa is extremely common. The majority of CF patients will be colonized with Pseudomonas aeruginosa by the time they're late adolescents. Staphylococcus aureus is more a problem with infections during childhood, but is also seen to persist later in life. Burkholderia cepacia is not nearly as common, but it is extremely nasty. So these are the biggies that people tend to study. We're interested in E. coli as a CF pathogen. E. coli is generally not considered to be really anything in CF. If it's isolated, it's considered to be a contaminant typically. But our collaborator, Dr. Barbara Kahl, has been looking at this in 2 study centers in Münster, Germany. So this is a total of 176 patients that were observed for anywhere from 6 to 90 months, with a median of a little over 5 years. Their sputum and throat swabs were screened for E. coli. These were the swabs that were obtained as part of normal practice. And there was some of the first little bit of results that I'm going to show you here has been published. Essentially, E. coli is actually being seen at a higher rate in this study population than has been shown previously. So a little over a quarter of these patients had E. coli isolated at least one visit. And not only that, but actually a little under half of those had long-term isolates. So this means that we had the same clone of E. coli being isolated at least twice over a more than 6-month period, indicating that this may be, in fact, colonization and persistence, not just, and they have a tendency to have E. coli show up as a contaminant. And what was really great is that, like a number of other CF pathogens, particularly Pseudomonas aeruginosa, which has been very well studied, we see a mucoid phenotype that emerges in these patient strains. So these strains are overproducing a capsular polysaccharide. They're thick, gluey, goopy, mucus-like. In Pseudomonas aeruginosa, this is shown to be associated with, sort of persistence, Pseudomonas aeruginosa has been shown to be a really common thing that crops up and actually is associated with worse patient outcomes in some studies. So what does this look like? In these patients, again, these strains are being isolated over at least the course of 6 months. With some patients, it's not for a particularly long time, but with other patients, E. coli is being isolated almost continuously over the course of many years. And you may even have 2 completely separate clones of E. coli within a patient on a couple of occasions. What we're working with is a set of isolates that was provided to us from Dr. Kahl from a patient that was designated patient O. These strains span a number of years, and they have both normal colony phenotype as well as that mucoid colony phenotype. And not just mucoidy once or twice, but both mucoid and normal isolates appearing to persist over time. And so these are, again, sort of blown up a little bit, isolates specifically from patient O. So multiple isolates obtained at certain time points and over the course of years. we picked two for closer examination. The first mucoid isolate and a normal isolate, normal colony phenotype isolate, that was actually obtained from the same throat swab. And these are designated 08 and 09, the 8th and 9th isolates from this patient. So, we did sequencing and analysis of these strains. First step, of course, Illumina sequencing, actually getting the whole genome sequence of these. We then used a suite of sort of scripts developed in the lab called pan-core-gen in order to identify the ORFs in these strains and compare them to each other, as well as compare them to our reference strains. This also identified the most likely 0809 differences. And with the help of Sandeep, we put together some scripts to sort of help us figure out which are more likely, which are less likely, because next generation sequencing is great, but at this point, it's still not perfect. So you're going to get some false positives. Then went in and actually aligned the reads and manually examined the quality of the differences. So is this something that's noise, or is this something that we really do think is in the sample set? And finally, Sanger sequencing of the likely differences, as well as a number of the unlikely differences. So the ones that in here were judged to probably not be true. And indeed, the ones that we judged to probably not be true when we Sanger sequenced were not present. The ones that we judged to be likely true when Sanger sequenced were present. So, one, we confirmed that our analysis pipeline does seem to be working reasonably. And we have a list of differences between these two strains that we can now examine to see which one is involved in mucoid. So let me backtrack for a second and talk just a little bit about the reference strains that we used. Because obviously, what reference strains you use for an analysis like this is going to have an effect on what you pull out. So in this case, because we're looking at an XPEC isolate and we're interested in XPEC traits, we want to use strains that are also from extraintestinal niches, and so are more likely to have a similar suite of genes that they're using to survive and thrive outside of the intestinal tract. So all three of these, UTI-89, CFT073, and NA114, are extraintestinally pathogenic. The strain UTI is the same sequence type as 08 and 09, so hopefully very close will allow us to compare those. And with pan-core-gen, all three of these reference genomes are actually used for cross-referencing. So all the ORFs in UTI-89 are used to blast CFT073 and NA114, as well as 08 and 09, to find new ORFs present in these genomes, and vice versa, so that we're trying to get the largest possible set of genes that we can. The first question, of course, is how alike are these strains? Are these actually really closely related and therefore useful for us to be comparing? By looking at gene content alone, they do seem to be extremely closely related. They share a very large number of ORFs in common, and more than they share with UTI-89, which is of the same sequence type, therefore we assume more closely related to them. And as we would sort of expect, they share the most genes with UTI, fewer with CFT073, and even fewer with NA114. So this does seem to confirm that these strains are closely related, but gene content alone certainly isn't sufficient. You can have the same gene be present, but have completely different sequences. So we really want to compare what's the actual nucleotide difference between these strains as much as possible. How do we compute that? The big answer is we have to look at the core genome. We can't compare strains when there's, they're only present in two strains and say anything about nucleotide diversity overall. So we have to look at the open reading frames that are shared by all 5 strains. And how alike are they? Very. We actually only see 5 SNPs between 08 and 09 versus 1200 with the one that's in the same ST, and tens of thousands with the ones that are in other sequence types. So yes, these two strains are extremely closely related, and in all likelihood, these most likely emerged from a single strain over the course of the infection. So the Occam's razor explanation of this is not that the patient got nearly identical strains that persisted in the lungs twice, but that one strain persisted and has then evolved these differences. So what's the overall variation that we see? We see 3 SNPs that are specific to 08. So remember, since we've got UTI 89, we can place these SNPs in terms of this is on 08, but not on UTI 89 and 09, versus on 09 only. As well as a 9 base pair deletion, which is an in-frame deletion of a predicted membrane protein. Also got a couple of SNPs that are present in 09, as well as a large deletion of 8 genes. I think it's particularly interesting that of all of these changes, these are in some way going to be changing the protein, deleting it entirely or changing its amino acid sequence. Of course, there's a question, okay, well, is this recombination? How can you be sure? In this case, the location of these differences is all along the chromosome. So it doesn't seem like this is the sort of thing where we can explain it with recombination. So again, sort of a reminder, the phenotype that we're interested in is this mucoid colony phenotype. When I went into the literature to learn more about this, I discovered that the study of E. coli mucoidy is sort of 2 things. You've got people saying, hey, there are some mucoid mutants of E. coli that we have generated in some fashion by treating them with a nasty chemical, by doing mutagenesis and looking for things. But it's all sort of saying, hey, in K12, in laboratory strains, which is certainly potentially interesting, but we're looking in a clinical case. And actually, most of these that are generated, like chemically, have a tendency to revert. Whereas 08 will persist to be mucoid for a very long time, and on basically every media that we've tried it on so far. In clinical isolates, which is more what we're used to, well, mucoid isolates of E. coli from cystic fibrosis have been seen for decades, 40 years now, and are continuing to be seen recently, but nobody's really looked at why are they mucoid, how are they mucoid, what's causing this. So, what of these changes is most likely to cause mucoid? Well, the one that sort of struck first off is MDOH, which is a truncation. So this chops the protein down to a little more than 1/3 of its original length. And when I did a literature search, it's been previously shown that in a K12 mutant using transposon mutagenesis, inactivating MDOH results in increased cholic acid production and a mucoid phenotype. Okay, so what is MDOH, other than something I found in the screen? So the name comes from membrane-derived oligosaccharide. So there's a group of genes, MDOH and MDOG, sort of their own little 2-gene operon, and the rest of them are separate, that make polyglucose that's then had membrane-derived phospholipids added to it. These are synthesized in response to osmotic stress and seem to allow the bacteria to sort of cope with this change. MDOH itself is 2.6 kb, 857 amino acids long. It has multiple transmembrane domains, and its job seems to be passing the polyglucose molecules out. So, obvious first question, does MDOH truncation actually associate with mucoid? Yes. Though, of course, remember, these are clonal strains. We're assuming that these blue strains are a lineage. So this isn't perhaps as useful. Though we did also sequence for all the other 08, 09 specific differences. And indeed, the strains with the MDOH truncation all share the same mutations as MDOH, including this non-mucoid one, which in the future might be really interesting to look at because this looks like a mucoid to non-mucoid revertant, something that's been seen in mucoid isolates of Pseudomonas aeruginosa. All right, so in terms of, we've got this, it's associated with mucoidy. The next question is, we have strains from 19 patients, a number of these patients with mucoid isolates. Well, is this the case in any other isolates from patients? So we sequenced MDOH, a number of different patients. This is all of the polymorphic positions in this 2.6 kD long protein. One thing that I did think was interesting is that within a single patient, we're not seeing any intra-patient variation in MDOH. This isn't a particularly, high mutation gene. The other thing is, we only saw 2 intra-patient differences. We saw the one we've already described in 08, and then in patient A, there's a completely separate SNP, which is also a stop mutation. So 2 separate mutations causing truncation of MDOH, both of them observed in mucoid isolates. The evidence is starting to look really strong. But of course, you can't really say anything until you do the wet work. So, complementing MDOH. This is the last in a long, long line of schema that we use to try and figure out how we can complement this gene. It could be because this gene is long, it could be because it has transmembrane regions, it could be because the bacteria just really aren't happy with this particular gene. But whatever the case, they really did not want to express it. The thing that finally worked was using topo-TA cloning. So inserting MDOH from patient O, either the wild type or the truncated, under the control of the lac promoter in this plasmid. And this actually, thankfully, did work nicely. So, looking at the mucoid strain 08, adding the vector control, we've still got a mucoid phenotype. So, that's great. We're not seeing any effect of the vector. We don't have to worry about that in the analysis. When we add in the wild type MDOH, do we get complementation? I feel like I should have a drum roll in here. The answer is yes, finally. We see this beautiful transition from a mucoid colony phenotype to a normal colony phenotype just by the addition of this gene. Because I've had so much trouble with this before, I was interested in if there were some dominant and negative effects of it, potentially. And the answer is no dominant negative effects. When I looked at a normal isolate from the same patient, adding a truncated MDOH, we don't see any effect. And adding more truncated MDOH to a mucoid strain, it's still mucoid. Nothing is particularly different. So MDOH truncation is the cause of mucoidy, and this is a recessive trait. All right, so yay, conclusions. MDOH truncation is associated with mucoidy in patient O. We observed an independent MDOH truncation in a mucoid strain from patient A, and complementation restores normal colony phenotype. So we've got this mucoid lineage, it's persisting in the patient. The question we always have to ask is why? Why is the mucoidy helpful? Well, it could be protection from the immune system, from other bacteria, such as secreted toxins or possibly cell-to-cell killing from antibiotics. It could just be that the biofilm itself helps the bacteria persist. I will mention that we did some research, so colonic acid overproduction has not been shown to protect from neutrophils. Secreted toxins of Pseudomonas aeruginosa, when I did the experiments, the mucoid strain was not any more resistant to the secreted toxins than a non-mucoid strain from the same patient. And the panel of antibiotics that I looked at, again, I didn't see a difference. So, persistence, better biofilm production, is looking, as a highly likely thing. And there's work that Katarina Ulrich, who went through our lab, did with Dr. Matt Parsak in his lab, looking at a wild-type non-mucoid strain from patient O and the mucoid strain 08, and looking at their biofilm formation. And when we look at the wild-type strain, it forms a biofilm. It's, 10 micrometers thick, fairly uniform, nothing too exciting. Whereas when we look at the mucoid strain, we're now seeing a biofilm that has this much more complex architecture, and at the maximum is much thicker. So this is a healthier biofilm that's going on, and possibly one that's better able to stick around in the CF lung and actually stay there. All right, so what can we conclude from all of this? From our genetic studies, there are at least 3 lineages that have emerged in patient O following persistent colonization by a single strain. These clonal isolates have persisted for years as both the mucoid and the non-mucoid lineages, and MVOH truncation is the cause of mucoidy and has occurred independently in 2 patients. So let's go back to the beginning. What was I talking about? What have I shown? So adaptation to the extraintestinal niche and looking at natural variation to explore the various ways that these bugs are able to persist and to adapt to new environments. First off, potentially pathoadaptive point mutations target the non-coding region upstream of the type 1 fimbrial adhesin gene, FimH, in X-Spec strains. The within clonal evolutionary analysis that I did gives us a clearer picture of the accumulation of pathoadaptive mutations over the short term. E. coli strains are evolving adaptively during long-term colonization of the CF patient lungs, and mutational inactivation of MVOH is indeed responsible for the emergence of the mucoid phenotype in E. coli colonizing its patients. At least some of them. Obviously, there are mucoid strains that we didn't see the MVOH truncation, so there's still a lot for us to look at and see what else is going on. So with that, I'd like to acknowledge all the people that made this possible. Dr. Sokurenko, of course, and all the other members of the lab, every one of which has helped me solve a problem at least a couple of points during my graduate school career, as well as our German collaborators who allowed us to access all of these strains, and Petra and Katerina particularly, who came to our lab and did a lot of interesting work with these strains. And of course my committee members for being with me through all of this, helping me out, reading drafts of my dissertation, giving me good critiques, and essentially making it possible for me to get here. And so, now the little bit of fun. So first off, the lab, our Caesar, and all the other fun that we've had. Again, these people have been great for me to work with. They've also been great people for me to actually have fun with. And again, as I told Sandeep earlier, Sandeep, you need to look at the camera.